Alright, so we're going to pick up now and look at our third section of the circulatory system part of this chapter, looking at the control over heart rate. And so as it turns out, it's kind of a baseline. Some people might be a bit higher, some people might be a bit lower, but as a baseline number, the sinoatrial node of the heart, which again was like the pacemaker of the heart, is going to help our heart maintain a approximately 100 beats per minute range of pulse. If you're at rest, so the 100 is sort of at a moderate level of activity. If you're walking down the street, your heart rate might be at 100 beats per minute. If you are at rest, if you're just sitting there, laying in bed reading or listening to a dull lecture, for example, the parasympathetic division of your autonomic nervous system is going to keep you in the range of around 70 beats per minute. So if you're at rest, if you're in good health, your heart rate should be down around 70. If you're exercising very heavily, you might increase it substantially. So you might get up in the range of 150 or 160 if you're exercising fairly strenuously. The other part of the body that can sort of be brought into play here would be the endocrine system, where if you release epinephrine, for example, that's going to cause an increase in heart rate as well under situations of stress. So what that's going to do is cause the cells of the SA node to fire more frequently, thereby leading to an overall increase in heart rate. So the next section we're going to look at is blood. Changing gears fairly substantially now, we're going to look at blood and take a look at some of the components of blood as well as what those components do. Blood, remember, is a special type of connective tissue. There are cells in the blood, erythrocytes, thrombocytes, leukocytes as well, kind of the three main types of cells we're going to look at. They're all surrounded by a matrix, which in this case is plasma. Plasma consists of primarily water with a bunch of other proteins like albumins and globulins and fibrinogens that help clotting and those sorts of things. The formed element part of the blood are the cells themselves. So the functions of blood would be transportation, and again, this would be everything from gases like O2 and CO2 to nutrients to waste and hormone and those kinds of things. It can be regulation of all kinds of things from pH to pressure to temperature to who knows what else. Lots of things regulated by the blood itself, but also protection because we're carrying, like I said, the parts of the blood that can lead to clotting if there's damage to the circulatory system, as well as immune cells that can help fight off any pathogens that might be introduced into the body. Those are also going to be carried in the blood and can help protect us accordingly. The plasma itself, if we're not looking at the blood cells, if we're just looking at the plasma is made up of about 90 to 92% water, so it's a relatively large component of water. There are things dissolved in the water, there are nutrients, there are breakdown products of digestion like amino acids and monosaccharide sugars and fatty acids and fairly simple molecules that have been broken down from the food that you eat. There could be waste products like urea, carbon dioxide. There could be hormones as well dissolved in the blood. There can be ions there, sodium ions, chloride ions, potassium ions especially can be in the blood. We see albumins involved here. Albumins are one of the solutes that help make sure that osmotic pressure is within the range of where it needs to be. So albumins can help either to, they can be regulated to either make sure there's more water in the blood or less water in the blood depending on the needs at the time. Globulins are involved in nutrients transport and immunity. Fibrinogen is the beginnings, the precursor molecule of something called fibrin, which can be involved with blood clotting as well. So lots of functions here in the blood. If we look at just the red blood cells, and again, red blood cells are also given the name erythrocytes, so when you see or hear erythrocyte, you think red blood cell. They are incredibly abundant in the body. There are about 5 million red blood cells per microliter of blood. So if you think about what a liter is, think about a 2-liter bottle of pop, and you cut that in half. In that 1 liter of blood, there are about 5 million blood cells times a million. So 1 million times 5 million red blood cells in that liter of blood. A microliter is 1/1,000,000th of a liter. So the other way to think about that is take your 1 liter of pop, divide that up into 1 million little subcomponents. In every single one of those little subcomponents, you're going to have about 5 million red blood cells. So they are incredibly abundant inside the body. If we look at the part of the blood that's occupied by red blood cells, it's also given the name hematocrit. And that's one of those numbers you might be given if you ever have blood work done. They'll actually give you a number for your hematocrit. One of the things unique about mammals and our red blood cells is that mammalian red blood cells do not have nuclei. You can see in this picture over here, these are very characteristic, normally shaped red blood cells. sometimes described as a biconcave disc, there is no nucleus inside that red blood cell at maturity. When the cells are being formed, when they're being produced by the bone marrow, they do have nuclei, but they go away during that maturation process. Our red blood cells also contain a protein called hemoglobin, which of course is kind of front and center when it comes time to transport oxygen or CO2 or other gases. Hemoglobin is that really important molecule that helps to transport those substances inside the blood. So it's a pigment molecule, it's got an iron in it, and it's very, very good at grabbing onto oxygen as well as other gases. So we'll take a look now at the structure of hemoglobin. A fairly complicated little protein here. It's made out of 4 protein chains. Every single one of these 4 protein chains, 1, 2 down here, 3, 4 up here. So the 2 red ones are the same as each other, the 2 blue ones are the same as each other. Every single one of those 4 chains has an iron, what's called a heme group, kind of front and center to each chain. And every single one of those heme groups is able to bind to oxygen in the lungs, and when that occurs, the hemoglobin actually goes through a physical change where it changes color. It goes from being sort of a dull, dark red to being a bright, bright red. Once that heme group has grabbed onto an oxygen, it actually changes color. When the blood is circulating, when it's going out to various parts of the body, it's of course going to be dropping off oxygen, not all of it. Remember, it only gives up a small fraction of the oxygen it actually carries, but once it drops off O2, it's also able to pick up CO2 that was produced by the cells and get that on out of the body at the lungs. The red blood cells itself are produced in the red bone marrow. We talked about how the nucleus is removed during maturation, and these guys have a lifespan of about 4 months. So even though they're relatively small, these cells live for about 4 months, during which time they play a huge role in keeping you supplied with oxygen, keeping CO2 from building up too much, supplying nutrients, allowing for all kinds of things to take place inside the blood. If you look at sort of the estimated number of red blood cells that die in your body, it's about 2 million red blood cells per second. So every second that you're alive, you're losing about 2 million red blood cells on average. That's a pretty humbling number to think about. That many cells dying at any given point in time. But the bone marrow, most of the time, does a pretty good job of replacing those red blood cells. There's also potential here to adjust the red blood cells. So if you were to move to a higher altitude, if you were to move to Denver, say, from Ohio, you're going to go from just barely above sea level to very far above sea level, and what will eventually happen is your bone marrow will start to produce more red blood cells to help your body get more oxygen out of an atmosphere that maybe contains a little bit less oxygen than what you're used to. So it's also adjustable. You can change the number of red blood cells that you have based on your environmental conditions. The blood cells that die off are going to be taken out by the liver, taken out by the spleen. They can be recycled. The iron is recycled in particular because it's a very, very valuable commodity that we don't necessarily get a lot of. So the iron is stripped out of the heme groups from the cells that have died and is put back into new cells that are being formed. The remnants of the red blood cells can also then be passed out of the body. We talked a second ago about how the number of red blood cells can be increased or decreased depending on the environmental conditions. We see here in this picture a primary trigger or stimulus of an oxygen deficiency. Maybe this happens when you move or go on vacation out to Colorado or something like that, higher elevation. There's going to be less oxygen in the body because there's less oxygen in the atmosphere. So what's going to happen then is that erythropoietin, which is a hormone produced by the kidneys, is going to tell the bone marrow to start producing more red blood cells. And the more red blood cells you have in circulation, the better your body is at carrying oxygen, and the better you're going to be at getting oxygen out of that atmosphere that is relatively low on oxygen to begin with. This is also kind of the basis for blood doping, right? So if you go and train at high elevation to help increase stamina, this is what you're doing. You're telling your kidneys to make erythropoietin to tell your bone marrow to make more red blood cells so that you can carry more O2. So then when you go after a few months of training at elevation, if you then go and play a game or go to a meet at sea level, you should have a competitive advantage because you have more red blood cells in your body relative to people who trained at sea level. Right? You've got more red blood cells, you can carry more oxygen, which means you're going to have better endurance and better stamina. That mode of training in most cases is fairly, it's considered to be legal. There are other ways of blood doping that are much more dangerous and also much more illegal in most instances. It's not something that most sanctioning bodies are very fond of seeing when they have athletes who do that sort of a thing. White blood cells are also called leukocytes, and they are the minority of cells in the blood. Less than about 1% of your blood cells are in fact leukocytes. They are much bigger than red blood cells. We can see them in contrast here, relatively small red blood cells versus relatively big, various different kinds of leukocytes. And they have nuclei. These cells are also able to get out of the blood and migrate out throughout the tissues, looking for bad guys they want to hunt down and consume. So we can see things like lymphocytes. Lymphocytes are involved in producing antibodies for immunity. So when you're sick, when you've been exposed to a pathogen, lymphocytes are involved or can be involved in producing antibodies that will then fight off that pathogen the next time your body sees it. Macrophages are relatively big, very generalized immune cells that will take out just about anybody that they find in the body that shouldn't be there. Not very specific, so they might not be as fast acting as a very specific antibody, for example, but they do function pretty well in keeping pathogens at bay most of the time. So a lot of the time, most of the time, you don't even realize you've been exposed to a pathogen because the white cells in your body take it out before it has a chance to cause an issue. These guys, again, they're mobile. They can actually migrate out of your circulatory system into surrounding tissues and phagocytize any pathogens that shouldn't be there. Platelets are involved with clotting. So if you have an injury of some sort, these are what's going to help that blood stop flowing out of your body if you've been injured, if things are working properly. These are also called thrombocytes, and what these are fragments of what are called a megakaryocyte. This is a very, very large, almost like mothership cell that is branching off or budding off platelets that then function in healing up or sealing off wounds if they might occur. Now, these platelets don't have a real long lifespan, whereas red blood cells might live for 4 months. Platelets only live for about 10 to 12 days, and so they're turned over even faster. But what they do, if an injury does occur, is that they're going to start to get trapped around the site of a wound. So down here in this picture, we have some sort of a wound taking place. Apparently, you've just been stabbed, I guess. So there's now a cut or a break in the blood vessel. So the platelets are going to start to aggregate around that wound site. You bleed for a little while, but relatively quickly, platelets start to kind of come in and seal off that site. There is a metabolic pathway here involved in producing thrombin from a precursor called prothrombin and fibrin from a molecule called fibrinogen, but only when you have thrombin present. So this pathway is producing an enzyme which allows a second enzyme to be made. And what fibrin does is it produces these really sticky kind of threads here. It kind of looks almost like a spider web or a net. And what that does, once that net is in place, it's going to help. Here's our little net here. That's going to help trap more platelets. It's going to help trap red blood cells. It's going to help trap white cells that can fight off infection if there's bacteria or something that was present on the object that impaled you. It's going to help get rid of those pathogens as well. So once we have that in place, we actually have now a plug, in a sense, of material that's going to keep blood from exiting where it's not supposed to exit. So that's kind of the progression of blood clotting. And in most cases, even for a decent wound, it happens pretty fast. It's not like it takes hours and hours and hours for this to happen. It happens within a few seconds in most cases. As long as you're producing thrombin and fibrin the way you're supposed to, it's going to seal off pretty well, pretty quickly. So that's kind of the pathway that we see here. We've got a break. Apparently, you've got a tack now that's in your skin. It's been a bad day. You got stabbed and you sit on a tack. You've got a break in the circulatory tissue here, so blood is leaking out. We see a pathway of platelets sticking here as almost like a plug. We see prothrombin making thrombin. We see fibrinogen making fibrin. All of those events together are helping to make a clot there, which is going to, stop bleeding for a pretty big wound in about 30 minutes. If you have a paper cut or something, you're looking at maybe 3 or 4 minutes before a clot forms. So of course it all depends on the scale of the wound, but in most cases it happens pretty quickly. The last thing that has to happen is that it has to go away. So once the healing process has taken place, then the body has to figure out a way to dissolve the clot safely so that pieces don't break off and lodge in a vessel somewhere, an artery somewhere where maybe you don't want things lodged, coronary artery or something in the brain, something like that. You don't want those bits of clot, or if it's on the outside of the body, scab, to get stuck somewhere else in the body. All right, so we're gonna cut off here and come back to talk about the different types of blood vessels, and then wrap up with a discussion of the lymphatic system and how that's involved with the circulatory system.